Hello and welcome, dear students. Today we are going to discuss about the topic balanced polymorphism. The main objectives of today's lecture are to know the basics of genetic polymorphism, to understand the relevance of genetic polymorphism to evolution, to know the concept of balanced polymorphism, to understand the evolution of balanced polymorphism, and to understand the various examples of balanced polymorphism. Dear students, let us first start with the introduction to genetics polymorphism. Genes are located on chromosomes and different versions of a gene are called alleles. The standard allele found in a population is referred to as the wild type allele. Most plants and animals have at least 2 copies of each chromosome, one inherited from each parent, that is one from male and one from female. The copies of the genes found on these homologous chromosomes may be identical for that gene and may carry 2 copies of one allele. In that case, the organism is called homozygous, or the copies of the genes found on these homologous chromosomes may be different, and in that case the organism is called heterozygous. Alleles differ from each other in their sequence of nucleotides, which may change the structure and function of the protein the gene codes for. Because of this, different alleles may have different effects on an organism's appearance or ability to survive. These effects can be helpful, harmful, or neutral without any change. Ecological genetics is the study of genetics in natural populations. Traits in a population can be observed and quantified to represent a species adapting to a changing environment. This contrasts with classical genetics, which works mostly on crosses between laboratory strains, and DNA sequence analysis, which studies genes at the molecular level. Most studies in ecological genetics begin with one or the other of the 2 most prominent patterns in nature, that is, the adaptation, the fit between an organism and its environment, or polymorphism, the maintenance of 2 or more phenotype or genotypic forms within a single population by natural selection. With the development of molecular ecology, the latter can be significantly extended to explore phenotypic differences between species in a phylogenetic context. This permits a more incisive investigation of the environments that favor adaptive divergence of phenotypes between species. Moreover, the same molecular methods useful in phylogenetics can often be modified to identify the genes or chromosomal regions responsible for those interspecific phenotypes. Although natural selection is the only evolutionary force that can account for adaptation, several evolutionary forces acting alone or in combination can sustain a genetic polymorphism, at least transiently. Genetic polymorphism is defined as variations among chromosomes, alleles, or other DNA sequences. Genetic polymorphisms, whether studied in the form of allozymes, RFLPs, mini and microsatellite variations, or DNA sequences, have become useful tools in a variety of research fields such as population genetics, evolutionary genetics, systematics and molecular phylogeny, human genetics, agricultural genetics, and forensics as well. Dear students, now let us discuss the relevance of genetic polymorphism to the evolution. Genetic polymorphism, as defined by E.B. Ford, is the occurrence together in the same locality of 2 or more discontinuous forms of a species in such proportions that the rarest of them cannot be maintained merely by recurrent mutation. Thus, the primary goal of the ecological genetist is to differentiate exactly how natural selection is acting to maintain a balanced polymorphism by the relative strength of opposing fitness effects acting on the different sexes at different stages in the life history, at different localities, or at different times in the lifetime of that organism. Genetic polymorphisms through multiple alleles at individual loci provide a mechanism to tag a gene or a piece of DNA, providing a powerful tool for a variety of investigations. These investigations cover a wide berth of objectives, including identification of genotypes in paternity and forensic studies, tracking the movement of individuals in field studies, mapping of quantitative loci affecting economical traits in plants and animals, or mapping of disease genes in humans, and evolutionary comparisons of DNA sequences and chromosome organization between related species. The uses of genetic polymorphisms are almost endless. Within a mere 50-year period, our picture of genetic variation in natural population has moved from near monomorphism to ubiquitous polymorphism in all organisms whose populations have not gone through severe bottlenecks in their recent evolutionary history. Dear students, let's now discuss the types of polymorphism, especially the transient versus balanced polymorphism. Genetic polymorphism is one or two or more variants of a particular DNA. Transient and balanced polymorphism are two types of polymorphism that arise due to two alternate alleles of a gene. In transient polymorphism, from the two alternate alleles of a given locus, one allele is progressively replaced by another. In balanced polymorphism, the two alleles are in equilibrium with each other. Both versions of the gene are maintained in the population. Transient and balanced polymorphism can lead to phenotypic change and may alter protein expression. The term balanced polymorphism was coined by one of the prominent ecological geneticists, E.B. Ford, who actually has divided selective polymorphism into two categories: the transient polymorphism and balanced polymorphism. Transient polymorphism occurs when there are two alleles in the gene pool and one allele is gradually replacing the other one. This is due to a strong environmental pressure causing directional selection to eliminate one allele from the gene pool. So transient polymorphism caused by a new favorable mutation in the process of displacing its ancestral allele by natural selection was considered unlikely because advantageous genes will usually have been already incorporated into the genetic constitution of the species, whereas balanced polymorphism, on the other hand, is a situation in which two different versions of a gene are maintained in a population of organisms because individuals carrying both the versions are better able to survive than those who have two copies of either version alone. So balanced polymorphism is a system of genes in which two alleles are maintained in stable equilibrium because the heterozygote is more fit than either of the two homozygotes. Hence, the difference between transient and balanced polymorphism is based on how the alleles behave. Dear students, let's now discuss the examples of balanced polymorphism. Balanced polymorphism is a stable polymorphism that can be maintained throughout inheritance. In this polymorphism, the organism will have both the alleles of the gene rather than having two copies of either version alone. Therefore, in a population, both versions of the gene will be maintained. Usually, a balanced polymorphism is maintained in the heterozygous condition. The superiority or heterozygotes over the homozygotes allows the maintenance of balanced polymorphism in population. This heterozygote superiority is also referred to as overdominance or single locus heterosis. Under balanced polymorphism, there is neither the fixation of allele nor the elimination of alleles. If the heterozygote of any mutation is fitter than the mutant homozygote and confers an adaptive advantage, then the mutation is maintained at an intermediate balanced frequency rather than being fixed. Most of the alleles for which balanced polymorphism is maintained is the human population are associated with certain diseases or pathogen resistance. Most human polymorphisms are neutral or slightly deleterious, but some genetic variation is advantageous and maintained in populations by balancing selection. If natural selection eliminates individuals with determinantal phenotypes from a population, then why do harmful mutant alleles persist in a gene pool? A disease can remain prevalent when heterozygotes have some other advantage over individuals who have two copies of the wild type allele. When carriers have advantages that allow a determinantal allele to persist in a population, balanced polymorphism is at the work at that time. So this form of polymorphism often entails heterozygosity for an inherited illness that protects against an infectious illness. Dear students, let us discuss various examples of balanced polymorphism. First one, cystic fibrosis is an example of balanced polymorphism. Cystic fibrosis, an autosomal recessive disorder most commonly found in Caucasian population, shows balanced polymorphism through heterozygote advantage to the fatal diarrheal disease cholera. It is caused by a mutation in the gene cystic fibrosis transmembrane conductance regulator, that is CFTR, which results in defective chloride channel. Presence of mutation in both copies of the CFTR gene in a person causes cystic fibrosis disease. Cystic fibrosis causes accumulation of mucus in the lungs and gastrointestinal organs, thus causing infection and other related defects. Cholera is caused by toxins secreted by the bacterium Vibrio cholerae in the small intestine. This toxin acts through intracellular guanine nucleotide binding protein, which consequently causes the release of cyclic AMP, which in turn induces chloride ion movement through chloride channel into the lumen of the gut. This process causes an osmotic loss of sodium ion and water into the lumen, resulting into watery diarrhea. In case of CFTR heterozygotes, where chloride ion channels are less functioning as compared to the normal homozygotes, the influx of ions into the gut lumen is reduced, thereby dropping the chance of diarrhea. This selective advantage of cystic fibrosis carries against cholera allows the maintenance of determinantal cystic fibrosis gene in high frequency by balanced polymorphism. Dear students, let's now discuss sickle cell anemia as an example of balanced polymorphism. One of the classical examples of balanced polymorphism in the human population is sickle cell gene polymorphism. Sickle cell anemia disease in Africans can be nicely explained by balanced polymorphism. Heterozygotes do not get anemia. They are also resistant to malaria. Hence, heterozygote advantage in sickle cell anemia is a result of balanced polymorphism. Sickle cell disease is caused by the single nucleotide mutation in the beta globulin gene, thus causing production of abnormal hemoglobin, resulting into sickle-shaped like red blood cells rather than the normal. The sickle-shaped red blood cells are fragile and clog the blood vessels, thus causing damage to organs. So severe anemia as well as heart failure. Heterozygous carriers or HbA, HbS genotype of sickle cell trait in a population possesses mixture of both normal erythrocytes having normal hemoglobin and sickle-shaped cells, while homozygous carriers with genotype HBS or HBS of sickle cell gene in the population have greater proportion of sickle cells in blood. So despite the deleterious character of homozygotes, the sickle mutation exists in human population since the heterozygotes possess selective advantage of being malaria resistant. Dear students, let's now discuss Tay-Sachs disease is an example of balanced polymorphism. Tay-Sachs disease is conferring advantage to heterozygotes from tuberculosis. Tay-Sachs disease is an autosomal recessive genetic disorder mostly occurring in children due to mutation in hexa gene, situated on chromosome number 15. Hexa gene codes for enzyme hexominidase A, an absence of which causes the progressive accumulation of GM2 gangliocytes in the nerve cells of brain, thus causing damage to the cells. The accumulation of gangliocytes in the cells causes deterioration of mental and physical abilities, ultimately leading to the death by the age of 4 to 5 years. Being an autosomal recessive disorder, the individual must acquire 2 Tay-Sachs alleles to exhibit the disease. That is, they must be homozygous for it. Tay-Sachs allele is known to be most prevalent in Eastern European people of Jewish descent, that is Ashkenazi Jews, who usually marry within their close relatives. Although being deleterious in nature, the allele is maintained in the population by its virtue of providing protection to heterozygotes from Mycobacterium tuberculosis, the causative agent of infectious disease tuberculosis, thus showing balanced polymorphism. Dear students, let's now discuss glucose 6-phosphate dehydrogenase deficiency as an example of balanced polymorphism. So, glucose 6-phosphate dehydrogenase deficiency is a sex-linked enzyme deficiency that affects 400 million people worldwide. It causes life-threatening hemolytic anemia in which red blood cells burst. However, it develops only under specific conditions: eating fava beans, inhaling certain types of pollen, taking certain drugs, or contracting certain infections. Studies on African children with severe malaria show that heterozygous females and hemizygous males for G6PD deficiency are underrepresented. This suggests that inheriting the enzyme deficiency gene somehow protects against malaria. The fact that G6PD deficiency is sex-linked introduces a possibility we do not see with sickle cell disease, which is autosomal recessive. Because both heterozygotes and hemizygotes are selected for the mutant allele should eventually predominate in a malaria-exposed population. However, this doesn't happen. There are still male hemizygous and female homozygous for the wild type allele. The reason again relates to natural selection. People with the enzyme deficiency, hemizygous males and homozygous females, are selected out of the population by the anemia. Therefore, natural selection acts in 2 directions on the hemizygous males, selecting for the mutant allele because it protects against malaria infection, yet selecting against it because an enzyme deficiency. This is the essence of balanced polymorphism. Dear students, other examples of balanced polymorphism in humans. The blood group system in human population also shows balanced polymorphism. The alleles for A, B, AB, and O blood groups do not show selective advantage over each other, and genetic frequencies remain constant from generation to generation for each population. In addition to this, there are also several other hematological diseases that include thalassemia and ovelocytosis, in which the homozygote hemoglobinopathy is balanced by the heterozygote advantage of protection from malaria. Dear students, examples of balanced polymorphism also exist in animals. Apart from human population, balanced polymorphism has also been known to be maintained in animals. Warfarin resistance in rats demonstrates one of the best illustrations of balanced polymorphism. Warfarin is an anticoagulant which is commercially used as rat poison. It inhibits the production of vitamin K dependent clotting factor and anticoagulant proteins by disruption of vitamin K synthesis pathway. So warfarin resistance in brown rats, that is Rattus norvegicus, is controlled by an autosomal dominant allele R. However, the resistant individuals also possess a low fitness due to altered vitamin K metabolism. The resistant homozygotes having two Rs, that is two dominant or capital R alleles, have 20-fold increased demand of vitamin K as compared to heterozygotes with genotype capital R and wild type susceptible S allele. Weird's demand of vitamin K is reduced to as much as 2 to 4-fold. The resistance allele R exists in balanced polymorphism in the population due to the higher fitness of heterozygotes, that is RS, over homozygotes, that is RR genotype and susceptible SS genotype. So homozygotes suffer loss of fitness as a result of vitamin K deficiency, whereas susceptible are affected due to warfarin poisoning. Further, we can think of an example. Let's consider two alleles of a gene that are responsible for producing enzymes that detoxify poisons and other chemicals. One allele has a higher activity of detoxifying chemicals, but it causes accumulation of harmful intermediates. The other allele has low activity, but the harm is less. Hence, the best situation for the organism is to have one copy of each allele. This is a type of balanced polymorphism. Dear students, at the end, let's discuss the evolution of balanced genetic polymorphism. Extreme genetic polymorphism maintained by balancing selection, that is so called because many alleles are maintained in a balance by a mechanism of rear allele advantage that is intimately associated with the important task of self-non-self discrimination. Widely disparate self-recognition system of plants, animals and fungi share several general features, including the maintenance of large numbers of alleles at relatively even frequency and persistence of this variation over very long time periods. Because the evolutionary dynamics of balanced polymorphism are very different from those of neutral genetic variations, data on balanced polymorphism has been used as a novel source for inference of the history of populations. Recent observations suggest that entirely novel selective forces may act in concert with balancing selection, and these novel forces may be extremely potent in shaping genetic variation at self-recognition loci. An important example of balanced polymorphism maintained by negative frequency dependent selection is plant self-incompatibility, a system for identification and rejection of self-pollen in or on the style. Self-incompatibility has arisen independently multiple times in the flowering plants, may involve one or more loci, and rejection of pollen may be determined by interaction between the parental genotypes, that is saprophytic self-incompatibility, or between the haploid pollen and the maternal diploid genotype, that is gametophytic self-incompatibility. The selection basis for the maintenance of polymorphism in these systems is readily understood from consideration of gametophytic single locus self-incompatibility. That is, if there is a match between the S locus genotype of the pollen and either of the alleles expressed in the style, fertilization is prevented. So rare alleles confer an advantage in encountering compatible matings within a population, so that their frequency tends to increase in succeeding generations. Conversely, an allele that becomes relatively common confers a relative disadvantage in encountering compatible mates. One simple distinction between balancing selection at the vertebrate MHC and the gametophytic S locus is that because selfing is prevented at the S locus, homozygotes do not normally occur, because the fitness benefit conferred by a particular incompatibility allele is a function of its frequency in the population, and different alleles are expected to be selectively equivalent. In contrast to the data for the vertebrate MHC polymorphism, the expectation of equal allele frequencies is reasonably well met in some but not all self-incompatibility systems. Similar frequency dependent selection is generated in mating compatibility systems in fungi. So heterozygote advantage of balanced polymorphism is important because it allows new mutations to be amplified in a population when the relative fitness of the mutant heterozygote is higher than the relative fitness of homozygote. So balancing selection results not only in the maintenance of a large number of alleles in populations, but also in greatly enhanced persistence of allelic diversity relative to neutral genetic variations. Dear students, it was all about today's lecture. Hope you have understood it well. See you next time with a new topic. Till then, take care. Bye bye.